So first off, let's talk a little bit more about lab. Actually, I'm kind of curious. How'd lab go yesterday? It's good. It's turned out like you were able to quality controlling, quality checking sequences didn't take like 5 hours.
No, that actually took like just a couple minutes.
Okay, that's good.
Alignments took a long time.
Yeah. Okay. So doing the different alignments and then looking at how they're different.
No, just getting it to actually line like
Oh, really?
an hour or more.
Half an hour. Wow. Wow.
That's more than most people.
Yeah.
Okay.
So
That's
that's
like
like
most
the-
people.
Yeah, I went to just like, I'm just gonna leave, come back. One of them I just quit, the window.
The copy one didn't work. I don't think anybody.
Wow.
But like mine actually like tried until like it was loading, and I was like, okay,
Yeah.
it'll be like the
It
last one.
tried
And
for
then, like,
most people, and they just.
This is good to know. Alignment took like a minute on my computer. I tried these. Like I went ahead and tried all of this stuff with one person's sequence. Like, hey, let's just grab some sequence and make sure this doesn't take insanely long. And like alignment took like a minute. So it's like, hey, you know, at the outside, I have kind of an old computer anyways that often frustrates me and makes me want to throw it out the window of my car driving 60 miles per hour. And at just the right time, so it smashed into the windshield of someone else's car and like totally goes into a million pieces. Then I feel kind of bad about the person's window, and then like especially if it had a kid riding in the front seat and glass went into their face, I feel really bad about that. Anyway, so I probably won't kill it that way when I finally do kill my laptop. But all that being said, I didn't think it was going to take a half an hour for people. But this is good. This is good to know. This informs me about something. Because what it means is we're going to be doing more processor, even more processor intensive stuff later down the road. Which means maybe a good strategy would be to have them install some of that software onto the lab or the computers in the computer lab, and people that have slow computers can go run the phylogenetic runs downstairs, because it will take, if that took you a half an hour, this will take you days. Like as it is, the runs as it is will probably take people about half an hour to maybe longer. Which is, I'm not exactly sure how to work this into a lab yet. There'll probably be like lots of like hanging around and like doing other things, or like leaving until your run is finalized. But yeah, it might take a while. So I will look into that. So thank you for that. Any other comments, questions, concerns? We'll talk a little bit more in general about it. Did you enjoy like poking around your own sequences? Is that kind of cool? Okay. Well, good. A couple things here. First off, some of you probably got weird results when you actually tried to idea how to that. At this point, that's expected. You probably won't get super accurate results with what we did yesterday. And there's a couple reasons for that. Number one, what you did is you actually only used half of your information. So in the next time we run this, it's not going to be next week, but the week after that, and the next time we run this, we're actually going to take and put together those two sequences that you got into one data set and then run that, and that's going to give you more information, which will give you more accurate results. Also, just taking a genetic distance and looking at the nearest thing is a good approximation, but it isn't definite. It doesn't give you, it's not the most rigorous way to compare this. And so, what ends up happening then is you get you get things that are slightly not correct. And that's because by looking at genetic distances, by themselves, we are just looking at essentially the number of changes and then possibly the likelihood of those changes depending on different kinds of changes. And essentially we're finding out how dissimilar these are. It's not looking at a most likely pathway to get from a common ancestor to where you are at. So, and that will place you differently. So, it might be that when you build, and this is the more rigorous way to do this is to construct a phylogenetic tree. And that's what we're going to be doing in a couple weeks, and that should get you better results. In fact, if you had all the same data set, about 50% of the time, maybe about 40% of the time, the phylogenetic tree will give you a different answer than just the genetic difference. Slightly different answer. However, that being said, since we're also changing the data set and expanding that, it'll probably be more than half of you will get different answers when you actually run the entire thing, your entire data set in a phylogenetic tree. And the reason this is because essentially all we're doing with a genetic distance is pairwise comparing every sequence and saying, how similar is this to this? How similar is this to this, and this to this, and then this to this one, and this to this one, and so on and so forth. And we're looking for just the smallest pairwise distance, which is great. When you run a phylogenetic tree, we're actually taking the context of all these together. And we're saying with the distribution of all of them together, what's the most likely arrangement so that we have some reasonable pattern of descent? So for instance, we have haplotype A, B, C, D, whatever, on to like X and Z, right? And I don't even think they're actually connected like this. But imagine that at first you came off, let's imagine another one here with H. You initially clade or like genetic distance, you're very close to D, but you're also very close to like maybe an H, or you're also very close to, but the differences are slight, and you're just ever so slightly more close to like a D haplotype, right? It could be as you run a phylogenetic sequence that, oh, this one is slightly more different. The H is slightly more different, but when we look at the changes, the number of changes in the context of everything, and we actually add up, you know, like, as we do this algorithm, we're going to talk more about this not next week but the following week. As we actually do the calculations for this, where instead of actually looking at distances, what we're interested in is the number of changes that we have to assume along this tree to get a particular arrangement. It will still probably be a fairly similar one that you're coming out with, but looking at the probability this way, which how many changes and how probable are those changes to make an arrangement that is most likely representative of descent, is going to give you a slightly different answer than just a pure genetic difference. Does that make sense? That will make more sense probably if it's still like, yeah, okay, whatever. It'll probably make more sense if we talk more about phylogenetics in a little while. And for right now, it's just kind of like, hold on, if you got wacky results, don't freak out right now. However, all that being said, you might still get results that you think are wacky even after you do that. In which case, there's another thing to consider here. So, HVR1 has a 50% probability. Okay, so these have known mutational rates. We have a good idea of what they are. HVR1 has a 50% chance of accumulating a single mutation, and this is just in the 500 that are HVR1 proper, and that stretch every 52 generations. HVR2 has a 50% chance of accumulating a single mutation and that's in the entire stretch. So what we're talking about then is this is the accumulated probability of a single mutation at every single base in there. And that has a 50% chance of accumulating a single in about 28 generations. So it's a lot. So this is yeah.
Wait, so it's out of all of them combined?
Yeah, this is all of them combined. So like
one to occur out of all.
yeah, in the entire sequence. Now in your HVR1s, you had something like, for instance, 1100. If you got the whole sequence, like 1100 bases, and the end got really bad, and so you had to chop it off. HVR1 proper, like the actual limits of that is about 550 bases inside there, and HVR2 is about, another 560 or something like that. And so it's 1/50th of this essentially is the mutational rate in each one, or 1/500th of that approximate mutational rate in each individual base. But here's the point. Is that even if you come up, if you think, okay, what is my descent, like, okay, I know what my mother was, I know what my grandmother was, I know my great-grandmother, her great-grandmother, maybe her, okay, you're still only getting back, like, a little ways in this. And so your matrilineal line, it's always possible, it takes a weird skew somewhere reasonably far back, that you don't know about. And because we're actually looking at fairly deep phylogeny when we look at this. So another thing to remember, if you come out with like the exact same sequence as someone else in class, that does not mean you are long lost siblings. That means that you are long lost cousins at like the 28th power or the 52nd power, something like that. You come from your, yeah. And another thing to kind of point out here is well, no, I won't point that out. Well, I mean, it's obvious. So mitochondrial DNA is passed clonally from mother to offspring. And so how we normally think about family trees, especially traditionally, has been through the father's line. That's how we track like names and other things like this. So like I actually have a book out there that actually follows my patrilineal line like that 13 generations of things. I know like really pretty exactly. Well, 13 generations in the United States and even farther than that further back. So I know where like my pretty decently where my name has come from and stuff like that, and that's how we have tracked like human lineages for a long time. And mitochondrial DNA is like going the complete opposite route in a way that we have not well context or haven't well followed through most of human history. So this often gives people very different answers than what you might otherwise think of. Anyway, so that's just a couple random things to throw out there when we're talking about this. And that'll come up more. Next week we're doing like structure prediction and things. I'll mention we'll have a guest lecture in. Does anyone have biophysics in here? Echo? Hey, we're gonna have Roy Campbell come talk about structure prediction. Yeah. I'm sorry, Ryan. Did that make you cough? I just immediately had one person clapping and one person coughing. I was like, wait, I don't know quite what that means. Yeah. I like Roy Campbell. Roy Campbell's a good person. But he also knows way more about sequence structure, especially in proteins. I think I might know a little bit more in like RNA. Yeah, proteins, so those. Anyways. Okay, so with that, let's go on to other things we're talking about. So I'm going to really briefly kind of recap what we're talking here about alignment. And so just again, you did this yesterday, and this is a recap from Wednesday. Alignment, this is anytime that we compare sequences, we need to align them, and so that we're looking at homologous bases. And as you probably figured out, all alignment algorithms are not created equally or do not create exactly the same alignments. There's small differences in there. So at some point we'll probably talk more about alignment selection and alignment algorithm selection, but not particularly here. And just alignment is we're getting these pairing homologous positions together. And essentially what we do is that program that you ran, or those algorithms that you ran, essentially calculates a likely or alignment score just as it is right then, and begins to introduce gaps to the sequences so that we get higher and higher alignment scores as we go along. And those gaps are just kind of null characters. We're not changing the actual sequence, we're just introducing spaces essentially in there so that we can get it to line up with other sequences. And then in this, the end result is then we have this matrix where each row is a different sequence, and then each column then has a homologous set of positions. So for instance, here would be a completely unaligned sequence, and then here we've introduced gaps into that sequence, and so now everything, you see they're fairly homologous, or appear to be fairly homologous going up. So here, stretch of 4 T's, and then we have a GA, and then a stretch of T's, and on such like that. Now, we can do this a couple different ways. We can do this just pairwise between two individual sequences, and one of the famous tools that does this, and does this actually fairly fast, which is BLAST, which stands for Basic Local Alignment Search Tool. And so let's just talk about BLAST just for a second. BLAST is another one of these tools. Here we go. BLAST is another one of these tools that is put out by the NIH and NCBI, and essentially all it does is you give it a sequence, and what it does is it then performs local alignments to essentially quick local alignments or quick. I could go more into the algorithm of this, but I'm not going to at the moment. Essentially, it performs very rudimentary alignments to lots of different things, and it starts with using very short sequences, like a set of 3 bases to eliminate a lot of things, and then expands that either way and starts eliminating other sequences very quickly until it arrives on several that are fairly close. And then what you get then is it will return a series of different sequences that are very similar to yours. And by similar, all we mean is percent identity, which we're going to talk about in just a few moments, which just means what percentage of bases are exactly the same once it aligns it. So I'm going to grab, let's blow HVRs. Who had a good one? Kalia, what number were you?
7.
Okay, let's go grab 7. HVR 1 for 7. Go, here's the sequence. Open that up. Open that up. You see, my computer is slow too, but it still only took a minute or so for alignment. Okay, I don't show tips anymore. I'm annoyed with you. Close. I'm just gonna take the middle here somewhere like that. Do a trim, and then you just put it in here, so you can just paste it into the box. You can also upload fastest sequences, so you see here, you can just upload your fastest sequence as well. And then there's a lot of different things that you can choose. We want to look at humans, and we're gonna blast this. You hit blast. And that thinks for a little while, and it's going ahead and it's doing pairwise alignments with a lot of other sequences. And it's going to come up with something. So I actually was reading a little bit about BLAST the other day because I was going to talk about it, and I realized someone said there's only like 200,000 queries per week. I was really surprised by that. That seems really few. Why aren't more people blasting things? I don't know. Which is an interesting comment in several different contexts. It is true in both reasons. Why aren't more people blasting things? What? That should still count as a request. That was a search. I want that to be counted. Okay, okay, I'm going to take out the human genomic. Okay, let's just like go back. So to get to BLAST, you can just search BLAST. Google search. And you can just click here, or if you're doing nucleotides, you can just click nucleotide blast down here, or a protein blast. You can also blast protein sequences. So let's go back to nucleotide blast, and that gets us to the same spot, I think. Okay, what in the world? I'm going to do a shorter section of your, apparently your wonky DNA here. Let's take that. Blast! I'm not going to change anything now. So now we're blasting against everything. Humans and rats and mice and elephants and whales, apparently. Someone misspelled whales in that data set. Oops. That wasn't me. I borrowed the data set. Literally whales. What's that? Well, it was spelled like actual. It was not spelled W-A-L-E-S. Like, it was W-H-A-L-E-S. Yeah. I think they meant W-A-L-E-S, but someone accidentally put the... Oh, here we go! Hey! And so this is what you get out. You get out a graphically kind of representing identity there is, which there's a lot. Who knew? You probably share a lot of homology with a lot of other human beings out there. Crazy, I know. And then you can kind of come down, and it gives you a sequence of these. Okay, so the first one that it comes out with is Homo sapiens sequence D loop, which is what this is in, complete sequence mitochondria. You match a human. What do you know? Actually, if you look down, you'll probably match a lot of humans. So actually, all I just searched for 1,027. Wait, no, the actual total alignment. 556 nucleotides. Yeah. So let's look at this one because this is also 100%. Actually, this is another one. Okay, so let's search this one. You are exactly the same as this. And what it does is it gives you that sequence then. It actually gives you, it reads out the alignment. And it also gives you a link to the sequence ID. So this is the accession number here. You can click on that. And actually get the information behind that. So hey, let's find some things out about you. Not about you, about this. Okay, so Source, mitochondria, human. Good to know. And the title of this was a complete mitochondrial control region for regional Caucasian population. Comments out of context are really kind of crazy in here. Okay. Good for you. And then we do another one. Okay, also had 100%. We can go look at this one. And probably you did because I just grabbed something in the middle. Hungarian mitochondrial population databases from Budapest and Baranya County, Roma.
This matches a lot better than yesterday's lab.
Yeah. Well, and this is yesterday's lab as well. And what I did is I just grabbed this. You can barely see it, but I grabbed from here to here just to make sure I wasn't getting anything on the outside that was crazy. So that probably is different. So if there's more, there could be more things outside of this that it's not matching up on that would change how it aligns. But here, that's actually another kind of good point here is that this is why quality control is really important. This is also generally why doing multiple sequences is really important as well, but we're not going to do multiple sequences for you, is that a single wrong base call in here will totally change your results. And this is why if you really want to have confidence in anything that you do, like when I'm publishing stuff on octopuses, are the coverage that we have on the museum specimens of octopuses that we are doing, I think we have like 10 to 13x coverage of those sequences. So we've done 10-fold PCR and sequencing for every base that we have called in there. And so we have a lot of confidence. On the ones that I'm doing from the modern ones, we don't have to do as much because it's recent. And so the coverage that we have on those is like 2 or 3. But still, we have independent reactions and sequences that confirm every single base call that we're calling along that sequence. We don't have the luxury of that here, so that might introduce a little bit of skew in there. Anyways, so that's basically how BLAST works, and then you can. It's really nice for identifying things, kind of a quick and dirty method, because you can get a quick look at, like, say you amplify some, and you might have done this if you're a micro, for instance, bacterial DNA. You don't know what bacterial species is. You can blast it and get a quick and dirty look at what it is, what it probably is. All that being said, again, just like our alignment and our distance, this is doing essentially the same thing that our alignment and distance was. It's just using a slightly different distance method. And so what happens then is you get error that's associated with that a phylogenetic approach will again give you a different answer than simply an alignment and a distance method. Yes?
What was your, like, did you run tests on your, yourself? And
So mine was a crappy sequence. I couldn't do anything. Yeah. So I was kind of sad about that. I was also gonna hand that out as like, if you have a crappy sequence, here, use my sequence instead of like going and grabbing one from someone else. And I couldn't do that either. So it's just really. I might see if I can salvage something from that, but yeah. And then there's multiple alignment. This is what you guys did last time, and it's more processor intensive than what you have otherwise. You might notice that I just pairwise align things with everything in the genetic, in essentially the NCBI GenBank database, and it was fairly fast. And you doing it on your computer was probably only comparably fast to another, 30 or 40 sequences? You would think it would be a lot faster on there. Well, there's two reasons for that. Number one, this is running not on your, when you run Blast, it's running on their computers, it's significantly faster than your computer. Number one. Number two, when you do multiple alignments, it's increasing not linearly, but exponentially the amount of processor time that it takes instead of doing pairwise, a bunch of pairwise sequences or alignments. And so, what we're doing here is it's an extension of pairwise, but we essentially do pairwise with each of them pairwise, and then each of them with each other pairwise, and then there is a way to get reconciliation between those. Anyways, long story short, it takes a long time, a lot longer. You're going to align many sequences to each other, and there's a lot of different algorithms for this process as you have seen in lab. They each have their strengths and weaknesses. They work differently well on different types of sequences. However, sequence alignment doesn't always work, and this doesn't always work especially when sequences are evolving rapidly. Because what happens is you can begin to lose signal in all of the essentially changes and bases that you get. It actually works really fairly well and fairly accurately and repeatedly on something like what you were doing, because most of the bases were the same between your sequence and all the other sequences. There's not a lot of positions that have changed. This is not always the case. There are some sequences or some distances between things in which they are so distantly related that there has been so much change that it's very hard to align. And how we do this at that point is we change our alignment strategy. So what we do is we use, instead of actually just using the sequence itself, we use the structure of whatever we're looking at to add additional information to help add to our alignment problems. So for instance, in proteins, we can actually look at the structure of the putative protein that this makes and look at different domains and then match those structurally to another similar protein, even if all the amino acids have changed. If the domains remain the same, we can align these domain for domain and see what amino acids are operating the same way and still get an alignment, even if we've had complete loss of signal inside the actual sequence itself. Now this is really useful, especially when we have, like for instance, probably very, very long divergence times between things, in which case, let me give you an example, and some of these can come out of you yourself as well. So for instance, you have both hemoglobin A and B, like the alpha and the beta subunits in your body, right? Okay. Those likely descended from one hemoglobin gene, some point. However, at this point, all mammals now have A and B. Your A is more similar to like a dog's alpha subunit. Your alpha subunit is more similar to a dog's alpha subunit than it is to your beta subunit. So there's a lot of divergence there, probably because those genes diverged further ago than yours and the dog's. Okay, so to align things like this, and this actually is common with a lot of things. You have had very homologous genes that have diverged for a very long time but still end up in you. And to align those, we can't use just the sequence information anymore. We have to align by structure. Another place this comes up, particularly in DNA, is if you're sequencing ribosomal RNA. So for instance, if you sequence 16S or 12S or something else ribosomal RNA, and a lot of these are used as markers for, like, 16S is used as like this very universal thing for bacteria. That's great. When you run this in BLAST, one of the things that introduces error when you simply blast those sequences is that secondary or RNA is a very rapidly evolving ribosomal RNA is a very rapidly evolving sequence. The reason for this is because most of it doesn't actually code for a gene of any kind. It's not making protein at all. It's the RNA transcript is the end working product that you're making from this. And it has its own structure to it. And so we generally need to align to that structure. What it has is, it has these places where the RNA actually comes back, folds on itself. Have you guys talked about this at some point in some class, like genetics? Okay, good stuff. It folds back on itself, and then you have binding of complementary pairs. So you have these pins, you have loops, and then they go up and they have this entire structure that then goes out like this, where you have an entire big structure of ribosomal RNA. It actually turns out that you can actually, especially in these loops, change genetic sequences very, very quickly, because there's not a lot of selective pressure maintaining the specific sequences in the loops. There's a little bit more in the pins because they have to bind complementary to other things. But what you'll notice is that there's actually places, like if you look right here, there's a non-complementary pair at that position in this particular ribosomal RNA. And that works pretty okay. It doesn't break the entire structure of the ribosomal RNA if you have a non-complementary pair in one place or another. If you have a lot of them, yes, they'll start breaking pins, but a few of them doesn't. And eventually what will happen is you'll end up what appears to be with a mutation on the other side that makes the other side complementary, and that pairs again, and you're all good again. So you can't accumulate too many non-binding pairs or non-complementary pairs in the pins, but you can accumulate some. And so there is some selective pressure on these to maintain kind of a conserved sequence, but not nearly as much as in coding sequences like coding for a protein, where if you get one base off, you can break the entire protein and then things die or things don't work very well. Ribosomal RNA is not that way. So what we have is we have this system that is evolving actually relatively rapidly, and we have lots of changes that build up between even you and I in ribosomal RNA, or us and other organisms, or between one type of bacteria and another. So when we align these, there are specific alignment programs that take into account this secondary structure. So actually, what is conserved is the particular loops and the pins in here. Those structures are conserved. And so what they do is, even if all the bases have totally turned over on completely different things, or completely different sequences, we still align this same loop with that same loop in another organism, this same pin with the same pin in another organism, and we take that secondary structure into account. And so this is actually really important, and if you use standard just standard alignment programs that just take into account the sequence with things like ribosomal RNA, if you can get really, really wrong results if things have diverged too much. They work kind of okay if you have fairly, like, if I do it with like some closely related octopuses in the same genus, or if you use it with humans, a bunch of humans, it'll probably work okay. Just a sequence alignment. But if you're dealing with things that are more distantly related than that, you need to take into account secondary structure when you're aligning ribosomal RNA and other things like that have a structure component to them. And then are also rapidly evolving, because again, the sequence signal can get totally obliterated, and the structure signal is really all you have left. And so again, when BLAST aligns ribosomal RNA to other RNAs to try to find a match, it's not taking this into account, and so that's one way in that just blasting like a 16S ribosomal RNA can sometimes it'll get you the things that are really close, and that's good. Like if you get a near exact match, but if you're dealing with something that's never been sequenced before and you're starting to try to look for okay, so this sequence has never come up, and I'm now down to like 95% identity that you're dealing with. All of a sudden, you can start getting really wrong answers with that. If you get exact hit, you're good. If you get stuff that's less than that, then there's errors that are introduced with that. Okay, let's talk about genetic distance, and this was the last thing that you did in lab yesterday. And essentially, again, as we're doing any comparisons between sequences, we need to align them before we can do this. And the genetic distance is now just a measure of the dissimilarity between sequences. So how similar or dissimilar, particularly with distance, we're looking at the percentage or the amount of dissimilarity between different sequences. And there's actually, you might think this is really simple, like we just count the number of distance or the number of differences, and that is one way to do it. There's actually a lot of different ways that this can be done. So for instance, we can just do what's called identity. This is one thing that is done. Then there's different models for actually calculating distance. There's Jukes, Cantor, 69. Well, here's something I'm going to talk about specifically. The Kimura 80 and Tamura and Nei 93. All the numbers on there are the dates of the publications that actually were these people describe these ways of calculating genetic distance. So let's talk about how these are different. So identity is just the proportion of sites that are different. This is the most simple. What percentage of the sites are exactly the same? What percentage are different? And identity is what percentage are the same. So if you have 100% identity, this is the exact same. And again, this is when you blast something, this is the type of genetic distance it's calculating, is just the identity. Pretty much just there. Now we're going to go into some slightly more complicated ones. The next one is Jukes Cantor 69, which is also just abbreviated JC69. And this assumes that the probability of all kinds of changes are equal. Now this is a distance. So it's calculated differently than identity, but it's going to give you essentially similar answers to identity because all changes are considered to be equally probable. So it's the same as essentially counting them up. Now it's calculated differently because this is a true distance. It's not, like identity is a percent similarity. This is going to be expressed in a distance, so it's going to be generally like between yours, if you calculate it this way, it's the number of differences. And again, this is, when we're talking about ways to actually calculate distance apart from identity, this is the simplest one. Slightly up from that is Kimura 80, or just K80. And this was described by the author named Kimura in 1980. And this is actually the distance that you used when you calculated in R. Again, in R, you can actually assign any of these that you want. And the default is to use Kimura 80. And this is also known as the Kimura 2-parameter model. And the difference here is that we know that transitions and transversions have different probabilities of occurring. So a change between an A to a G and a T to a C tend to be more probable than a transversion, which is A to a C, A to a T, C to a G, or a G to a T. So essentially, if we're going from purine, or pyrimidine to pyrimidine, if we're staying in the same class of base, that's a different likelihood to happen than transversion where you're changing the type of base that you have. So Kimura 80 actually takes that into account when we are looking at the distances between sequences. And this is why, for instance, BLAST will give you a different answer than a genetic distance that you calculate with like something Kimura 80 or something like that. And then finally, Tamura and Nei 1993, or just TN93, is a 3-parameter model in which each type of transition has a different probability. So an A to a G and a C to a T each have their own probability. And then all transversions, that should be transversions down below, not transitions. All transversions have the same probability. Any questions on genetic distance so far? Yeah.
I know this is slightly different, but when you're running the alignment test, there, you know, they always pull up a box where we had the option of changing, basically the probability of how it aligned for each different, codon. And what type of situation would you actually use that, or could you justify setting your own parameters for that?
Changing your own parameters. If you have different, if you know there's different kind of processes working, like for instance, sometimes different parameters work on, say, coding DNA versus like non-coding or regulatory DNA versus ribosomal RNA versus things like that. Generally, I have very rarely ever been in the position to really tweak with the defaults on most of those before. Yeah. So now, all that being said, I think in a future lab, depending on how much time we have, I will actually bring up situations in which we want to do that when we align for the phylogenetic tree. Any other questions? Well, with that, that is all I have for today. So enjoy your weekend. I'll see you in here on Monday. We'll begin talking about secondary structure or structure prediction.